Hey guys, and welcome to another FRQ practice. We've had our official Google Meet meeting at 11 a.m. this morning, and we talked about a couple of FRQs that were data-based. Right now, I'm just going to go ahead and go over those, what we talked about, a couple of the tips and tricks we discussed, and how to solve these to better help you guys prepare for your digital exam. Let's go ahead and get started. So first, we're going to do a quick read through. We're going to underline any pertinent information that we see as coming out as important, such as numbers or references to variables or any of the sort of main themes of the experiment or something else. And then we're going to go ahead and try to put that together into a quick analysis of the experiment. So let's begin. The common bedbug is a species of insect that is becoming increasingly resistant to insecticides. Uh-oh. Bedbugs possess several genes suspected of contributing to the resistance, including P450, ABC8, kind of a funny name, and CPS. To investigate the role of these genes in insecticide resistance, researchers deleted one or more of these genes in differing strains of bedbugs, as indicated in Figure 1, and treated the strains with the insecticide beta-syphlothrin. Each strain was genetically identical except for the deleted genes and was equally fit in the absence of beta-syphlothrin. The percent survival of each strain following beta-syphlothrin treatment is shown in Figure 1. Now, what have I underlined? So when I was reading this through to the participants in the live session, I underlined resistant to insecticides because this whole entire study is about resistance or resilience to insecticides. These bed bugs have some sort of adaptation that makes it so that those insecticides don't work very well with them. I underlined it because it's a central theme or idea that I was picking up on, and I wanted to make sure I kept that in mind as I went through. I then underlined each of the specific names of genes they gave me because I figured that those would be important to know and to take note of as we went along. P450, ABC8, and CPS. I then underlined the name of the insecticide, and then I underlined this because this helps me to better understand how they're defining their variables. Each strain was genetically identical except for the deleted genes. So that sounds like a control to me, or some sort of way that they're trying to set up this experiment. So basically, I went through and I didn't underline everything. I underlined the names of the variables or whatever it is that we're working with. I underlined the main idea. I underlined the thing that the other thing we're working with, insecticide, and I underlined something that told me something about how they're setting up their variables. So my first thought whenever I come into an FRQ should be, what experiment are they doing? What is their scientific question? What are their variables? What are they trying to do? Because even though it gives me that information in this paragraph, it's giving it to me in a very technical language that I might not be able to instantly translate in my brain very well. So I want to kind of try to break this down a little bit. The first question that I want to ask is, what's their scientific question? What are they trying to figure out? So I want you to take just a couple of seconds to take a look at this intro and pause the video and see if you can come up with their scientific question. What are they trying to figure out? If you had to condense everything you just read right here into one question that these researchers are using to drive their study, what would that be? Okay, hopefully you pause your video. This is what we came up with. What genes help to provide resistance to insecticide in bedbugs? So we came up with that because we realized that it said that they were deleting one or more of these genes, in which we could rightfully probably underline that as well. So they were deleting one or more of the genes to see how that affected the bedbug's ability to survive beta-syphlothrin, or whatever it's called. So that's the question we came up with. What genes help to provide resistance to insecticide in bedbugs? So we decided then that the variables we were working with was the IV were the deleted genes. That's what the researchers were changing. The DV was insecticide resistance, or the resistance of the bedbugs to the insecticide. And then even if you wanted to put in a control, if you wanted to, you could even add that the control is that each group is genetically identical, save for which genes are deleted. And that's obviously control, because if they weren't genetically identical, then we could expect maybe some of the differences in their genes besides what we change, what we delete, might be causing the trends that we're seeing. So we're trying to get rid of any extraneous variables or bias in the data here. So let's go ahead and take a look at the graph. So the next thing we want to do is a graph. And notice I still haven't gotten about to answering this question. The reason why is that it's more important for me to understand in a database FRQ what the story of the data is, what they're trying to tell me, than the content knowledge or the question they're going to ask me. Because I guarantee they're going to ask you about all the stuff that we're doing right now. And I promise you, it's like one of those Mr. Miyagi moments from The Karate Kid. You just got to trust that what I'm showing you right now is important and pertinent and a good idea. And you'll see it come to fruition in a little bit. If you don't get that reference, please watch The Karate Kid, either the old original or the new update, as both are fun to see and are, I don't know, some cool lessons in there. Anyway, so let's take a look at this graph. So we said that our independent variable was the genes, which ones were deleted and which ones weren't, and sure enough, now this is interesting because they had 3 different genes they were looking at and they wanted to put it on the same graph. They went ahead and just made a chart that stands in for your x-axis. So here's all the different strains of bed bugs, and here are the genes that are either activated, so they're still there, that is indicated by plus symbol, or they've been deleted. And they did the right thing. They deleted them one at a time and left the other 2 active to see how it affected the percent survival of bed bugs. And we can imagine that the higher the survival rate, the higher their resistance or resilience to the pesticide, the lower their survival rate, and the more the insecticide worked on them. So if we're looking at this right now, I mean, my first question is, which gene seems to be the most important? Well, it seems to me, and we all sort of decided this, that ABC8 was most important. So if you look at P450 getting knocked out, well, yeah, it did reduce it a little bit, quite a bit actually, and these error bars do not overlap, which means that there is a statistically significant difference between the two. So yeah, getting rid of P450 definitely decreased their ability to survive against the insecticide, but good God, look what happened when we knocked out ABC8. Wow, that went down. Now it's somewhat comparable to the change that we see over here. It's not statistically significantly different than P450 based off these error bars alone because it overlaps, but it's definitely significantly different than the error bar and the bar over here with just the normal wild type control. So man, getting rid of ABC8, big difference. And here we don't see a big difference between the CPS. This error bar overlaps with this one, so I mean, honestly, this is not statistically significant in terms of survival versus this one. So getting rid of just CPS alone isn't going to do it. And then getting rid of these 2 at the same time, wow, that combo, they must be working together in some kind of way because that combo is really killing these insects. So what I've basically done is I've just gone through it, I've read through the graph, and I've identified for myself the trends. And this is sort of what we came up with. This is what we came up with before. And it seems we said that ABC8 seems to be the most impactful gene for resistance overall, whereas P450 and ABC8, when they're deleted at the same time, the resistance decreases a lot, and the deletion of CPS alone doesn't seem to be significantly different than the wild types. Now, this took me maybe a minute or 2 to figure out, and I know you have 25 minutes to figure out your FRQ, which is going to follow this exact pattern, by the way. It's going to give you a scenario and data, and then it's going to be ABCD. But I've only spent a couple of minutes doing this, and let me tell you, I've already done most of the groundwork for this FRQ. So let's go ahead and dive into what I mean by that with part A. So after I've explained it to myself and I have a pretty firm grasp of what's happening with the situation, I'm going to go down here to part A. It's going to say, explain how the deletion of a gene can change the fitness of an organism. Now I'll tell you that part A in each of these is going to require a little bit of content knowledge. Now for most of these FRQs, you can actually solve them without content knowledge. Part A will be a little content primer to be like, hey, do you remember this from your studies? Now, so far in our classroom, we're just about to talk about this in the next week here and preparing for the exam and just the course in general because we started with evolution, so we're a bit behind because they cut evolution. Ha ha ha, isn't that funny? From the exam, but it's going to be fine. We'll get to it. But right now you might not completely understand this. However, you should remember that a gene is a segment of DNA that codes for a protein, and that protein determines your traits. And so if we were to delete a gene from your body, a bit of DNA instruction from your body, I mean, what do you think would happen? One of your traits would be deleted or it would be severely affected. And if that trait was something that was going to help you survive in the environment and it got deleted, well, guess what? You're probably not going to survive very well. And so that's what we came up with here. When a gene is deleted, the DNA instruction is deleted, and the traits based on those instructions aren't going to work properly. If those traits were important to the organism's survival and reproduction, its fitness would decrease. So that organism would not be doing well. That's pretty much common sense. You can fairly figure that out even if you don't really remember all of the content. Another thing to point out, though, is that these part A's are always going to be very low point value. And it's 1 point here for this. So, hey, you missed 1 point, oh well. If you can't remember the content, don't dwell on it, move on, because you're going to be delightfully surprised by part B, which says identify the dependent variable and the independent variable in the experiment. Ah, man, see, if only we had done that in the beginning. See what I mean? Okay, so part B is basically exactly what we've already done. So we've already identified the dependent variable and the independent variable, racking up 2 points. Woo! And then we just got to justify the need for each strain to be genetically identical, which we've already talked about a little bit. So we went ahead and just kind of copied and pasted. Now, the thing about the answer, you can't answer it like this in the exam, and I'm so sorry, but I should have done this better. But so this was our question. We don't really need this in the answer here. That's kind of unnecessary. But instead of just being able to go IV, deleted genes, DV, insecticide resistance, or whatever, we actually can't do that. We have to write it in full sentences. So you could say the independent variable is the genes that were that were deleted were kept wild type. Okay, and I think an abbreviation like DV would be fine. You might want to type it out just to make sure. But in the DV is the percent of bed bugs that survive the insecticide. Now you could have said was insecticide resistance, and I think that would have counted just as well because, I mean, if you're surviving, you're resistant to the insecticide. But I'm just using the official AXI title that is here for the y-axis. Oops. Okay, and then we said if the strains were not all genetically identical, then the results of the study might have been affected by other variables besides the IV introducing bias into the experiment. We've already said that. We already talked about it way back at the beginning, and boom, part B is done. So basically, in our setup, we did part B, which is amazing. Which leads us to part C. Using the means and confidence intervals, justify the claim that ABC8 is effective at providing resistance to beta-syflithrin. Well, guess what? We've already done this too, because we pointed out that this error bar does not intersect with this one at all, and in fact is farther away from intersecting with this one than this one over here. And so we can comfortably say that ABC8's error bar provides the strongest significance and does not overlap with the control wild type than any of the others that were knocked out on their own. So what we said was the error bar of ABC8 is significantly lower than that of the wild type, showing that there's a significant difference between the two. ABC8 has a most significant drop in survival when deleted, and I guess you could say, for clarification, versus the other genes. So wow, that was pretty quick. We didn't really have to do very much with that, and we're good to go. So that's part C in a nutshell, and boom, this then more points. Awesome, and so then we get to part D, which is always going to be the hardest one. So basically the difficulty is supposed to ramp up as you go along. So part A is just a background knowledge check. Part B is like, all right, can you identify the variables? Can you, do you understand experimental design? Part C is like, all right, now we want you to kind of explain things a bit more. And part D is where they're like, okay, we want you to really put things together. So part D is going to be the tricky one. And so if you find yourself spending most of your time on any part of the FRQ, you should probably spend a lot of it on part D. P450 encodes an enzyme that detoxifies insecticides. ABC8 encodes a transporter protein that pumps insecticides out of cells. CPS encodes an external structure protein located in the exoskeleton that greatly reduces the absorption of insecticides. Based on this information, data make predictions that explain how deletion of 450 and ABC8 results in lower survival in bedbugs compared with the deletion of CPS only, and justify. Right, so the first thing I'm going to say is you want to split this bad boy up because this is a big thing. So I'm just going to start in the very beginning. I want to clarify to myself which each of these genes are, because when I was reading through them, I don't know about you, but I super zoned out. So P450 encodes an enzyme that detoxifies insecticides. How could I say that in an easier way? Well, basically, detoxify gets rid of the toxin. I could say P450 diffuses the insecticide or the poison. That's exactly what I said. P450 diffuses poison. Haha. Okay. So I know what that thing does. It gets rid of it stops poison from being poisonous. That's cool. ABC8 encodes a transporter protein that pumps insecticides out of the cell. So basically, ABC8 gets rid of insecticide. And I think when I said ABC pumps the poison out of the cell. Cool. So P450, yep, it goes ahead and diffuses it, and this pumps it out. That's really cool. Now I clarified both of those for me. CPS, what does it do? It says it encodes internal blah, 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 blah. Basically, if I'm reading this right, CPS is going to reduce the amount of poison that the cell can actually take in. So it reduces the absorption of poison. That's cool. Okay, so now that I've clarified to myself what these guys mean without all this fancy technical language, I can keep going. Based on the information in the data, make predictions to explain how the deletion of P450 and ABC8 results in lower survival. So here's what I'd recommend doing. I'm going to just type out exactly what I just said, okay, just for my own sake. This is not necessarily the answer, just me trying to figure it out. P450, okay, P450 diffuses the poison. All right, cool. ABC8 pumps the poison out of the cell. Okay, so now it's the first thing it's saying how a deletion of P450 and ABC would result in lower survival in bed bugs. Okay, cool. Well, I'm going to say, okay, what would happen if P450 was broken? All right, so if P450 was broken, oh my God, was broken. Ah, okay, if P450 was broken, um, poison would still be active in the cell, so it's dangerous. Okay, if ABC8 was broken, the cell would be unable to pump the active poison out, I was doing so well, of the cell. Okay, so that's the reason why that would really suck. That's to compare it with the deletion of CPS only, okay? So I'm just going to say here, just continue my thoughts, if both were broken, the cell would be full, or I would say be trapped with a bunch of deadly poison. Okay, if only CPS was broken, now let me kind of go back and resume what I was thinking about with CPS. I said that CPS reduces the absorption, doesn't stop it, it just reduces the absorption effectively. If only CPS was broken, more poison could get into the cell. But if P450 and ABC8 were still active, they could diffuse and pump the poison out, oh my God, out of the cell. And boom, we're done. All I did, I didn't have any content. I have no idea what these genes are. I don't even have to know what a gene is. All you need to really know is that the gene does something. They help something. Or it encodes something, whatever that means. I don't even have to really understand the process. All I need to do was figure out the P450 diffuses stuff, ABC pumps it out. If both were broken, the cell would be trapped with a bunch of poison in the cell. If CPS was broken, more poison would be able to get into the cell, but if 450 and ABC were still around, heck, why does it matter? So that's why a deletion of CPS only is not nearly as bad as P450 and ABC. I have made predictions, I have justified my response, and I am good to go. And that's all it takes, guys. This was our original answer. So you can pause the video and take a look at it if you want to, but it's basically the one we just did. And that's it. And wow, look at this. So my first FRQ in the test, it gives me 25 minutes to solve it. And it took me, with all this explanation and everything, it took me longer. I mean, I'm up to like, what, 18 minutes right now? Okay, wow. I've got time to spare. That doesn't mean I should waste it. God, that's not bad. And that's going to be the format of the FRQ you'll see on the test. So pretty cool. All right, let's move on to the second one. So bacteria cultured in media with carefully controlled nutrient composition. A lot of C's in there. The graph above shows the growth of bacterial population in the medium with limiting amounts of nutrients, two nutrients, 1 and 2. Great. Okay, so if I'm looking at this right, I'm looking at the growth of bacterial population is what we're looking at in the presence of two nutrients. So here's nutrient 1, here's nutrient 2. Bacterial population is going up, so I'm guessing it's munching on the nutrients, doing pretty well. Here I can see it's population density. Now, if you're wondering what density means, it just means the number of things in a particular space. If I have a higher density, I have more things in one space than I would if I had a lower density, which means fewer things in the same space. So, if I'm talking about my hometown of Farmville, Virginia, woo, Farm Vegas, everyone, in case you don't know, that's what we call it. I don't know why. It's as far from Las Vegas, I imagine, as you possibly get. Not just geographically either. Farmville, if I was talking about the density, population density in Farmville, I would say it is far less probably than in Newport News. I mean, almost. There's more people per square footage in Newport News than there are in Farmville, trust me. All right, so that's what we mean by population density, how many little bacteria there are, and then here we see the relative concentration of nutrients are changing as well. So if I'm looking at this board and look at the problem, what's going on? Well, I can see the nutrients going down. That's obvious because they're being consumed, and the bacteria going up. Yay, they're growing because they're melting. That's neat. Okay, what else did I notice? Well, nutrient 1 starts going down immediately, whereas nutrient 2 doesn't go down until, God, what is that, hour 7 or something like that? Yeah, wow. So nutrient 1 is being munched on right away, whereas the bacteria kind of holding off on munching on nutrient 2. And why is that? I mean, I could be that they just prefer nutrient 1 to nutrient 2. It could be that nutrient 1 is like necessary somehow for them to be able to even digest nutrient 2. Maybe nutrient 1 actually blocks them from eating nutrient 2 until they're done with nutrient 1, and then they can kind of process nutrient 2. Maybe there's some sort of mumbo jumbo going on there. That's all the guesses I could have. Basically, that's sort of what I'm seeing. Nutrients going down, bacteria going up. Cool. All right, now that I have kind of an idea, these are kind of the scientific questions I came up with. They might have been asking, how is the speed of growth affected by nutrients 1 and 2? How are nutrients 1 and 2 affected by the speed of bacterial growth? How fast can bacterial population grow in limited number of nutrients? All these are good questions to ask, and could be sort of what this is based off of, better define what they're looking for us. The amount of nutrients 1 and 2, or really this is it's the relative concentration or the change in concentration is going to be, that's actually going to be the DV here. This is where I think I messed a little bit when I was going through it. I was reading. So our independent variable here is going to be measured over time. Really, I think time would be the safest answer here. Bacterial population density and nutrient concentration should be what we have for our DV. So I apologize for those of you who were in the live session. I think I did that a little bit incorrectly here. Anyway, so then nutrient 1 steadily decreases from the beginning. Yep, nutrient 2 starts decreasing about 7 hours. Nutrient 1 is first choice. Necessary. That was our guess based off of what we saw. So basically, we just did exactly what I just said. Looked over it, came up with a guess, our IV and DV and all that good stuff. So then, part A. Here's our background content knowledge. Describe how nutrient 1 most likely regulates the genes metabolism of nutrient 2. Now, again, we haven't really talked about gene regulation. But what does the word regulate mean? It means to control. Okay, so and metabolism, of course, breaking down nutrients into their chemical processes, how you into their chemical components, so you can make energy and stuff like that, like ATP and cell respiration. Metabolism is what you do with food, right? You get energy. Okay, so if we're going to describe how nutrient 1 controls our breakdown of nutrient 2, well, if I was assuming that nutrient 1 controlled it, I would say nutrient 1 is actually stopping nutrient 2 from being digested until nutrient 1 is gone, and then nutrient 2 can be digested. Or the other idea we came up with was nutrient 1 was slowly kind of building up the bacteria's ability to digest nutrient 2, and it wasn't until it reached a threshold that nutrient 2 could be digested. Either way, nutrient 1 prepares the body to break down the nutrient 2, or it could be preventing from breaking down nutrient 2 until all nutrient 1 is gone. This is a pretty safe answer, and I didn't even have to mention much about gene regulation, but I could just use the graph to kind of say, well, if I assumed one was under control of the other, that's kind of the pattern that I'm seeing. Didn't take any concept knowledge, all it took was me kind of looking at the graph and trying to figure stuff out. B, identify the dependent variable. Oh no, if only we had done that and identify one control of the research could use to improve the ability of the experiment. Well, we already said the dependent variable was going to be the bacterial population density and the relative concentration of the nutrient. We already have that right here, so we can just plug that in, make sure it's used. Full sentences, please, no less, remember. And then what's the controls I could use? Well, we grow both of these bacteria in the presence of both of these nutrients. So what if we just grow it in the presence of nutrient 1, and then again in the presence of nutrient 2? That would be really interesting to see, so we can have some controls to kind of compare to. Other suggested ideas for students was to introduce a third nutrient that we would grow to compare. That could sort of mess up with the IV, and that might be a little bit closer to a new experiment. But okay, maybe I could definitely see the merit in it. And then the third one, I don't remember the third one. Oh, we could have done with a non-edible medium, so we could have grown the bacteria in a medium without nutrient content and see how it would have grown. That would have been interesting. All three of these could have been interesting controls for various things, but I think they're all pretty good. Okay, so then let's see. What is the relation between the bacterial population density and the concentration of nutrients 1 and 2? Before I read on any further, I'm just going to tackle that first question. What is the relationship between population density of bacteria and the concentration of nutrients 1 and 2? Well, as I said before, As the nutrients go down, population of bacteria seems to go up. So I'm going to go ahead and say for that one, as the nutrient concentration decreases, bacterial population density increases. However, I did notice something interesting. When the nutrients are totally gone, bacteria population growth seems to plateau, seems to stabilize. That makes a lot of sense. Heck, if you don't have a lot of food, you're not going to do a lot of growing either. So basically, the bacteria population goes up and hits this plateau where we run out of food, and right then, bam, look at this, bacteria population stagnates and starts growing again as soon as nutrient 2 starts to be used, and then at the end of nutrient 2, boom, we get that same stagnation again. So just for it to be safe, I did a little bit of a comment there that was like, okay, when the nutrients depleted, the bacterial population density stops growing. Okay, here we have growth. Sorry. Second one. Based on the figure, calculate the growth rate in cells per milliliter times hour of the bacterial population between hours 2 and 4. It says calculate the growth rate. Okay, what is growth rate? What does rate mean? Well, rate is just growth over time, right? Or distance over time, or whatever. Whatever you're trying to measure. Like if I was trying to measure how fast I could run, let's see, I did, let's say I ran 10 feet in 2 seconds. Okay, so I'm just going to do this really quick on the calculator. Let's say I ran 10 feet in 2 seconds, if only. Okay, right? And so if I divided 10 by 2, 10 feet divided by 2 seconds, I would get 5 feet per second. That's my rate. So rate's super easy, guys. So we go in here to growth over time, and it says between hours 2 and 4. I'm going to go back to the graph and look at hours 2. Here's hours 2 and 4, and I'm going to follow the graph up. And it looks like at hour 4, I had 10 to the 4th was my population density, and at hours 1, or excuse me, at hours 2, I had 10 to the 1. So if I take 10 to the 4 minus 10 to the 1, that's going to be 10 to the 3. In case you were, wondering how I did that or something, if you can check this if you really want to. If you're wondering what 10 to the 4th means or exponents or something like that, 10 to the 4th is going to equal 10,000. If you don't know why that is, you could just do it's 10 to the 4th, so it's going to be 10 times 10 times 10 times 10, and you can see, yep, that's 10,000. A quick life hack here is that the 4 is going to tell you how many zeros come after the 1. So 1, 0, 0, 0, 0. Okay, 4 zeros, 10 to the 4th. Okay, and that is going to be minus 10 to the 1, which is just 10, in case you were wondering. So, yay. Minus 10 to the 1, and that's going to give us this. Now, this is where I also messed up before when I was doing this. I'm so sorry, guys. When I was doing the live session, I completely messed this up. So, 10 to the 4th minus 10 to the 1 is not 10 to the 3rd. God, sometimes I just really, really disappointed with myself sometimes. So this gives us 9,990. Okay, so 9,990 is what we get. Okay, cool. That's how much we changed in terms of bacterial population density. And what do we divide that by? Well, we had that over a 2-hour period, right? So from 1 to, so divided by 2, and that's going to give us 4,995. And what are our units going to be? Well, our units, and let me go ahead and change it here so you can see I did the stupid thing. Whoops. Whoa, no, that was part 4, sorry. I did the stupid thing and did 10 to the 3 right here. Let's go ahead and do this. So 4,000, what was it, 495 or 995. All right, cells per milliliter. Those were the units they gave us. I should put cells, shouldn't I? Not CLL. Cells per milliliter. What was it? Per hour. Yep, because we divided by 2 hours. Hours was our time unit. So there we go. 4,995 cells per milliliter per hour, which is pretty awesome. Okay, and then what prevents the further growth of the bacterial population after 9 hours? Well, if we go back, we can see at 9 hours we run out of nutrient 2. We have no more nutrients left, and so the population growth is going to stagnate, and that's what we put. No more nutrients left, so bacterial growth stagnates or stops right there. So hopefully that helped you guys with that. Again, it just took a little quick calculation and a little bit of reduced idiocy from the part of your teacher, who had a massive brain fart and made a horrifyingly simple calculation error, which definitely probably led to some misconceptions. So I apologize for that. Hopefully this helps. Okay, all right, so we go into the last part. Identify the preferred nutrient source of the bacteria in the culture over the course of the experiment. Well, we already did that way back at the beginning. We said nutrient 1 because it started munching on it from the beginning. Here's the graph to justify your response with evidence. Okay, well, we can see from time 0, nutrient 1 started going down, whereas nutrient 2 did not start until about the 7th hour to go down. So that's a big thing. We can also see that the general, well, I don't know. That's enough for me, honestly. I was going to say something about the rate, but this is a more precipitous decline, so maybe not. Maybe they're getting desperate at this point. Who knows? But anyway, that's enough for me. So go ahead and say nutrient 1 was the bacteria's favorite. Nutrient 1 was consumed for the start of the experiment. Nutrient 2 wasn't consumed until all of nutrient 1 was gone. So that's going to go ahead and answer that. Predict one reason the population does not grow between hours 5 and 6 and justify your prediction. Well, between hours 5 and 6, so here's hours 5 and 6. Why doesn't it grow? Because it's run out of nutrient 1 and it's not processing nutrient 2 for whatever reason. Either because nutrient 1 is it doesn't have enough of the nutrient 1 to process nutrient 2, or because nutrient 1 needed to go away completely before nutrient 1 could be processed, or the bacteria just really didn't prefer nutrient 2 and were waiting around to see if any nutrient 1 would come back until they decided, hey, what the heck, let's go with nutrient 2. So bacteria ran out of nutrient 1, could not or did not want to consume it, so growth stagnated. Their growth rate stopped right after nutrient 1 ran out and didn't pick up again until nutrient 2 started to be consumed. So all of this, it comes back to this, all of this I could get from this, okay? I could just read this thing, and it's just telling me its story all day long, and I can answer parts A, B, C, and D to the best of my ability and get points from them, okay? So content knowledge. I know a lot of you guys think you're, or I don't know if you think this, but a lot of AP students think I'm deficient in content knowledge, and that's going to be the real big problem. But the real big thing about this is not content knowledge, is what the other AP teachers are helping me understand. It's all about your logic and your reasoning. Are you able to take the graph, read it, pick it apart, figure out what it's trying to tell you, and figure out that story? That is going to be the key skill for this FRQ, this first FRQ you're going to see on the test. So I hope this helped you guys. Again, I apologize for calculation errors. Sometimes we all make stupid mistakes. You know what? It's great because I get to learn from it and take a little slice of humble pie, which is always awesome. It helps to combat the continual slide towards arrogance, which is definitely not what any of us want. All right, guys, I hope you have a wonderful day. Man, it's great. It looks kind of nice outside, so hopefully you're able to enjoy it. It's a little bit cold, but go outside, have some fun today, give yourself some time, but make sure you reach out to me if you have questions, concerns, whatever. I'm here for you, and man, I miss you guys, but it's been really great meeting you on the Google Meet. Make sure you can catch up with me there if you can. All right, guys, have a great day.